Hello students, myself Popere KS, Assistant Professor at Shikshan Sanstha's RBNB College, Srirampur. In this lecture, we are going to see the next topic, that is the from plant biotechnology. Now the topic is basic techniques of plant tissue culture. So in last lecture we have discussed about the introductory part of the plant tissue culture, what are the main bases on which the plant tissue culture work. So now today basic techniques of the plant tissue culture. So what are the various techniques which are used in plant tissue culture for generation of a new plant, or we can say regeneration of a complete plant. So here are some techniques. So first is callus culture. What is callus culture? Callus can be called as an unorganized mass of a cell. Unorganized mass of a cell, if it is kept on a medium, so it will start, if we kept a cell on a medium, it will start to grow and it will grow in an unorganized manner. So that can be called as a callus. And what is callus culture? It is production and maintenance of unorganized mass of a proliferated cell from isolated plant cell tissue or organ by growing them on an artificial medium under controlled aseptic conditions. So what we are doing in the case of the callus culture, taking the cell and allow, that is a plant cell, and allow it to grow on a nutrient medium, and aseptic conditions are also required for it to grow. And how it is going to grow? It will be growing in an unorganized mass of a proliferated cell, the cell which have the capacity to divide. So they will be continuously dividing, but they are not going to form any structure or any part of a plant. That structure is an unorganized mass of a cell. So first is callus culture. Now next is organ culture. Like callus culture, only callus is forming, it doesn't have any identity. But in case of the that is organ culture, there is a architecture is maintained of a particular organ, or we can say a complete plant can be grown. So as in case of the callus culture, no, it is an unorganized. But now in case of the organ culture, we can say it is an organized, or the division will take is an in an organized manner. So it can be called as differentiation and preservation of the architecture. As I have already told, it will maintain the architecture of a that is a cell, particular cell or an part, and it is in vitro culture and maintenance of excised organ of whole plant or part of an organ. So here it is maintaining the architecture of a particular that can be a cell, tissue or organ, or even an complete plant. So in this culturing, we can culture not only a particular cell but also a group of tissues or some particular organ, a complete organ can be cultured. That's why it is called as an organ culture. And the things which we can use for organ culture can be embryo, that is ovary or anther. So these are the things which can be cultured or which can be regrow by organ culture. Now next is shoot tip or meristem culture. Now what is meristem? Meristems are the region where cells are continuously dividing, or we can also call them as a meristematic cell. The cell which have capacity, the area of a, we can say the part of a plant where there will be continuous division of cell are going to take place, that are called as meristem. So what is a shoot tip of meristem culture? Now what we are going to do in this technique, that we are going to take the particular that is meristematic region or the shoot tip. So at the shoot tip there are regions that are called as SAM, that is SAM or shoot apical meristem. So these are the meristematic region where cells are continuously dividing, that is at the SAM. So that part, or we can say the tip of a tip culture is also called as tip culture. So the tip of the particular plant will be taken, that is containing the area, that is SAM, shoot apical meristem, and now it will be allowed to grow on a culture medium, that is on our medium. And this is also we can take the axillary bud or adventitious bud. So as these are the region where continuous division is taking, so we are going to take the axillary bud or the adventitious bird and then allow it to grow on a medium. So now from that particular that particular part, now the complete part or the complete organ or specific part can be grown. So this is called as a shoot or the meristem culture, because we are taking the meristematic cells, the continuously dividing cells, to grow and complete plant. Now after that, next is single cell culture. So now what will be happening in single cell culture, as in case of the callus or tissue, this organ culture or shoot, we are taking the group of cells, or in case of the single cell culture, what we are going to do, it is a method of growing a isolated single cell that is aseptically on a nutrient medium under control condition. So the name itself is telling we are only going to take a single cell and allow it to grow on a nutrient condition, that also under a control condition, or we can say the aseptic conditions are maintained. And how this aseptic conditions are maintained? By in the PTC, or we can say plant tissue culture lab, there all the things are particularly maintained so that the culture, why we are saying that it is aseptic condition, because we are not allowed to get it contaminated, so contamination is not allowed. Now next is suspension culture. Now what is suspension culture? Now when we prepare the media can be prepared in two forms, that when it gets set, it will be converted into a solid form, or sometime the media, they are in a liquefied form, or we can say in a suspension form, not in a liquid particularly, but in a suspension form. So that are called as suspension culture. So what happen in case of the suspension culture, there a single cell or a group or aggregate of a cell multiply by suspended in agitated liquid medium. So there the media, it will be somewhat semi-solid or suspension like, so it is continuously moving. In that case, aeration or the aeration is good. So this suspension cultures are used, and in suspension culture we can take a cell or a group of cell and allow it to grow under the aseptic conditions. Now what are the things which can be grown by suspension culture? First is induction of somatic embryos and shoot. So next is production of secondary metabolites, then in vitro mutagenesis, then selection of mutants, and last one is genetic transformation. So when we have to, our aim is to get all this thing, that is somatic embryo induction, production of secondary metabolite, genetic transformation, selection of mutants, we will allow this to do on a suspension culture, that is in a suspended, where the media is suspended or it is in a suspension form. So here the growth will be faster, because the aeration will be good at this suspension culture. Now next is anther culture. Now what is anther culture? Anther is a male reproductive part of a flower. So what is done in the anther culture? In vitro development of haploid plant originating from the potent pollen grain through a series of cell division and differentiation. So what is happening in anther culture? We are taking the anther, that is from the flower, and allow it to grow on a medium. Now here mostly it is done to produce a haploid plant. When we have to grow a haploid plant, at that time anther culture is used. And now how it will be going to grow? Through a series of cell division and differentiation. A potent pollen from anther, it consists of a pollen. So in anther there are number of pollens present in the anther. So here we are directly taking the anther and allow it to grow on the medium, that is on the nutrient medium. So the anther containing the potent pollen, it will be grow into an haploid plant cell. So that is anther culture. Now next is pollen culture. Now what is happening in polar culture? As in case of the anther culture, we are taking the complete anther and it is consisting of number of pollens. But in case of the pollen culture, what is happening? We are means anther is broken, it is and from that pollens are taken, and particularly single pollen is used, or that is anther is consisting of pollen. So particular pollen is taken and it is allowed to grow on a that is on a nutrient medium. So in vitro technique, which pollen grains are squeezed from the intact anther and culture of the nutrient medium in order to obtain a new plant. So what is done here? The squeezing of the that is anther is done, and from that pollen is taken out and it is allowed to grow on a nutrient medium in order to obtain a new plant. So but in case of the anther culture, there may be a chance that we can't get a pure haploid plant, but by pollen culture the results are that we get a pure haploid plant. So the probability of getting a haploid plant by pollen culture is very high as compared to we see through the anther culture. So we should use this pollen culture technique if we want to produce an haploid plant. Now next is embryo culture. Now embryo culture, like the other cases, here we are going to culture an embryo. So, and how this it is aseptic isolation of embryo from the bulk of the seed or capsule, and in vitro culture under aseptic and controlled physical condition on nutrient medium. So in this case, what we are going to take, we are going to dissect out or get out the embryo from the mature seed and allow it to grow in a nutrient medium. So from this now embryo, again a new plant can be regenerated. So regeneration of a new plant from the embryo, it is called as an embryo culture. Now next is protoplast culture. So what is protoplast? A cell without a cell wall is called as a protoplast. Okay, now what is done in protoplast culture? Here, culture of isolated protoplast, which is potentially capable of cell wall regenerating, cell division, growth and plant regeneration on a nutrient medium under aseptic conditions. So here that is a cell without a cell wall is taken and it is allowed to grow on a nutrient medium under aseptic condition, and now the cell, as we have removed the cell wall, but it should be potent, that is potent, or we can say potentially capable of growing a cell wall, that is regeneration, and also the further division which was required for the regeneration. So it is isolated protoplast and then culturing it on a nutrient medium, and it will undergo cell division, growth and plant regeneration. So complete plant can be again grow by this protoplast culture technique. So we have seen the different techniques, that is callus culture, organ culture, then that is stem culture or the meristem culture, now protoplast culture, single cell culture, suspension culture, embryo culture, anther culture and pollen culture. So these are the different types of culture techniques which are utilized or which are there for to obtain the complete plant or part of a plant, tissues, organ. So different techniques are utilized in plant tissue culture to obtain the different various kinds of results. Okay, so this is all about, this is just the basic technique of the plant tissue culture. Next in next lecture we are going to study how the media preparation and how the other processes will take place. Thank you.